to the organizers for organizing this course, and I am sorry not to be in person able to give it this lecture. Currently, there's what I call a sea change or a culture change in functional neuroimaging that's ongoing towards computation-rich open science sharing and mining of increasing stores of analysis-ready data. The trend in time series neuroimaging, including EEG, MEG, intracranial EEG, is to record small experimental studies with perhaps 20 subjects, and then to publish a single metric applied to those studies, an ERP or a spectrum or something, and then essentially to discard the data and start again on another experiment. This has been the pattern in academic laboratories in psychology, neuroscience, neurology. But there's a new pattern emerging, and that is for either for large studies, including lots and lots of subjects, or natural cognition studies in which the data are highly complex. Subjects watching movies, subjects performing physical exercise, interaction between participants. Here, there's a new pattern emerging. That is to propose, hopefully fund, and record the data, then publish not only papers on it, but data papers that document its inclusion in public archives. If those data are well shared in an analysis-ready format, then this can lead to a whole string of publications on the data and citations to the data paper, at least, if not collaborations with the authors. This is currently of most interest in departments called data science, bioengineering, biophysicists. And it will take a generation before the skill sets of these different approaches merge. Currently, there are two opposing trends in neuroimaging. One is the movement toward firm, valid, robust statistical testing of hypotheses declared in advance on uniform, reproducible data, with the goal of reproducing results obtained typically on small data sets. Quite the opposite trend is to be excited about applying machine learning modeling across large collections of data and perhaps of diverse data, with the example of the large language models at hand, of course. Why should data authors, either authors of small studies or large studies, want to publish analysis-ready data and make the effort to do so? Well, first, there is a satisfaction in contributing to science, to being of use to mankind, which I don't discount. Then there's the possibility that you could yourself reuse the data, or assign a student to do additional analysis on the data. If it's an analysis-ready format, then it's much easier for you and for them to do that. Analysis-ready data more readily supports collaborations and interactions among researchers concerned with the same data. The coin of the realm in academic positions is our citations. And data paper citations for well-appreciated, well-annotated data shared publicly on in some format will increasingly be recognized as a valuable contribution to the academic science enterprise. And finally, contributing to data archives is also allows you to enter your data into new directions in data modeling associated with compute facilities that are increasingly being tied to large collections of data, allowing new tools to be used without serious programming effort. The archive I'm associated with is funded by the National Institute of Mental Health under the BRAIN Initiative in the US. It's NAMAR. NAMAR.org is a front end to data that's been shared through the OpenNeuro .org archive. NAMAR serves the electrophysiology data, the EEG, MEG, and IEG data. It offers search, quick downloads, and visualization of the data, as well as data quality estimates. And currently has almost 300 data sets, totaling almost 30 terabytes of data from almost 14,000 participants, increasing steadily. From looking at all the data that have been, all these data sets that have been contributed to Open Neuro and are now served by NAMAR, we've come to realize that there's a strong gap in understanding of data authors as to what constitutes analysis-ready data. The current standards, BIDS and NWB, perhaps most frequently, are cross-platform formatting standards, and they serve and mandate submission of data with minimum high-level annotation, metadata, telling who collected the data, how did they collect it, on what machine, when, and where, and so on. But there has been no common standard for recording what happened during the experiment. No common standard. And I'm here to discuss with my colleagues the HED, or HEAD standard, which is the only standard that's been proposed and developed to do exactly this. So analysis-ready shared data must also include detailed, unambiguous event descriptions in a common language and syntax. It should enable data search and analysis based on details of the participants' experienced events, actions, tasks, task conditions, or on details of their data, of their brain data that have been added to the record post hoc, most likely. If it has all these things, it must also enable computing on these events, not only searching for them, but finding contrast in these events considered in the context in which they occur. So the HEAD, or HED system of a hierarchical event descriptors, when combined with a high-level formatting standards, such as BIDS, now provides practical path to sharing of analysis-ready data. Now, the recording standard in time series recordings for what happened during the experiment is only to write in a very sparse event log like this. Here, something what came on at 145 seconds, and what happened was event type 127. Here, they didn't bother to put in even how long that event lasted. as BIDS in particular does not mandate it. This is known in hundreds of labs around the world as a trigger. This is event log. This is one trigger in an event log. I'll get to why it's called a trigger later. But what does event type 127 mean? Does it have any universal meaning? No, it doesn't. For motor action events, typically in the past, button presses, there's only a time of switch closure that's actually recorded. It's considered to have no duration at all. When did this all start? Well, it started with Alfred Volkmann and then Wilhelm Wundt, the psychophysiologists in Germany in the second half of the 19th century, who invented the tachistoscope that allowed visual stimuli at first to be presented all of a sudden, and then removed all of a sudden, and allowed and then a falling weight on a string that allowed them to record how long it took the subject to press a response button. So for the next 150 years, reaction time was the queen of experimental psychology. And it's still treated in psychology as an instantaneous event, a decision event, ignoring the fact that the event of deciding to press the button to perform the neuromuscular signaling to that effect, to contract the muscles, to move the finger, to produce a switch closure, and then further phases is itself the brain-body event that in neuroimaging, we would like to understand how the brain supports this event in all its phases. Well, how did the term trigger come about? There was this was the stimulator that my mentor had in the first EEG lab I went into, the lab of Bob Glambos at UCSD, in which he had a actual grass stimulus generator, as you see here, and you could dial in to produce one beep every 1.2 seconds, and it should last this long and should have this frequency and this rise time and decay time. It would produce sounds, and then we'd record on a computer. This was actually way before my time. This was the first ERP computer, or it was called the EP computer at that time, the evoked potential. It was an analog computer that would allow adding up of traces following some set of events that were signaled by triggers from the T-tail pulses coming out of the parallel port, which would trigger a new collection, a new addition of an epoch in the computer. So these are the triggers. And this is still the conception underlying recording of events in neuroimaging experiments. Even in fancy modern EEG, fMRI, MEG systems. Okay, what are the 5 core concepts of the head system for annotating what happened during neuroimaging experiments? The most basic is the experiment timeline. There's a single timeline for all data recorded during the experiment, and all events are referred to that timeline. Events themselves are processes that unfold through time. They have to have a fixed beginning and a fixed end, which are marked in the record by event markers: onset marker, offset marker. They may also have marked time points within their event process marked. These may be points of interest or moments of phase transition. For instance, the transition from one syllable to the next in a spoken word. It's also important to include in HEAD the experiment condition events. These may be task conditions, they may be stimulus conditions. They may overlap, there may be a hierarchy of them. Together, these stimulus presentation events, participant action events, or data feature events that may be added to the record after the recording. All these can be analyzed with respect to the context in which they occur. Now, to learn more about this concept of events and recording what happened, Kay and I have just published a paper in Frontiers. You can consult it. It's open. So now I'd like to introduce Kay Robbins from the University of Texas, San Antonio, who is the technical lead of the HED working group. The HEAD working group meets twice a week. It's open, and she will give the structure of the HEAD system.
Hi, my name is Kay Robbins. I'm a member of the HEAD working group, and I'm going to be talking about HEAD, which are hierarchical event descriptors. I'll talk about concepts and tools. So HEAD is a standardization effort to move beyond keywords and triggers to do fine-grained analysis-ready fair annotation. We're entirely open source, and all our tools, documentation, and sample data sets are available online through our web page or through GitHub. So my talk is going to be organized into 4 parts. I'll talk about HEAD annotation basics. That is what a HEAD annotation is, what terms you're allowed to use, how you get started, what you do if there are terms you need that aren't there, and some motivation about why using a common vocabulary and structure are important. And I'll talk about HEAD's role in bids and what the relationship is between event files and sidecars. And I'll move to HEAD models of events and experimental contexts and why that's important and how HEAD support makes context available. And finally, I'll briefly talk about how HEAD fits into the brief picture of scientific workflow. So let's begin by talking about what a HEAD annotation is. HEAD annotations are comma-separated lists of head tags like sensory event, visual presentation, or cue. They come from specified vocabulary, which is organized in a tree structure called a head schema. And you can reference a head tag by its path, like sensory event, sensory event, or just by the tag itself, sensory event, which is the usual way. Each node is a subclass of its parent, so sensory event is an event. And this is important because it means that searches for event will also return sensory event. It's not just a text search. HEAD annotations can be sentence-like and use parentheses to enforce structure like the subject predicate of a sentence. And if you look at the overall structure of the HEAD standard schema, you'll see that there are 3 categories of terms. They're noun-like ones, like event, agent, and item. They're verb-like ones like action, and then they're description ones like property or relation. So if you want to look at the HEAD vocabularies that are available, we have a standard browser, which is online, and you can pick a schema version. And the bold items that are displayed are leaf nodes. And you can hover over items to get information or use this pull-down search bar to find things that start with whatever you type. So as an exercise, I encourage you to explore the HEAD schema, which is available at www.headtags.org. Now let's turn to how you actually get started doing annotations. And so we see here a table of terms that I want to annotate. They're in laboratory jargon, so they're not understandable unless you're familiar with the lab experiment itself. And we have a basic recipe for getting started on this. And the key thing is to first identify what kind of event we have, and then to add supporting tags. So that's the general strategy. And even identifying the event type, it is an important step in making your data more understandable. So HEAD only has 7 event types, and the most important ones are sensory event and agent action. And then the supporting tags. So for sensory event, you might want to say what kind of modality was used. Is it a visual presentation or an audio presentation or both? What kind of task role it has? If there are items in this as part of the sensory event, like a display of something, what they are, and how they play a role in the task. And then you might want to give some attributes. And so there's similar suggestions for the other kinds of events. So, for example, this non-WM is actually a cue. It's a sensory event. It's a visual presentation. Its role is instructional. And it's a dash. That's a black dash. Correct is actually a feedback. It's an auditory presentation. And you can group tags to, again, reflect that sentence structure that we mentioned before. So as an exercise, you might try your hand at annotating an experimental stimulus consisting of a green triangle and a red square presented for 0.5 seconds. So you see the basic idea, and the standard schema contains the terms that you usually will need to annotate an experimental timeline for a typical experiment. But you can also extend the schema in most places. For example, we don't have star as an item in the schema, but we do have 2D shape. So if you give the parent, you can say 2D shape / star, and as an annotation, which is more specific than provided in the head schema. You can also develop a library schema to standardize the terms in your subfield, and we're encouraging people who have communities with specialized terminology in their subfield to form a group and to standardize that field, and we'd be happy to assist you in adding it as part of the head ecosystem. So you can see on the right that you can attach these library schema terms to an appropriate place in the standard schema, or you can have some of them be independent subtrees. And there's, as I mentioned, there are a number of the library schemas that have been developed or underdeveloped. There's a MOBI library schema under development. There's also a score schema, which is in its second release, which incorporates into HEAD the score standard for clinical neurology annotation of EEG. So one of the questions that people often ask is why a schema and not an ontology? And the reason is that the schema view, which is it's a sub-tag system, it's a top-down view, it's a natural way for annotators and analysts to tag complex events in their data. However, and of course they can add library schemas to do more specialized terms. But HEAD is embedded in an ontology, so there's a larger HEAD information space, and that ontology is available downstream to link tools and which have a power of doing more provenance and semantics. But that structure, which is complex, is hidden from the annotators and the analysts, so they can just deal with annotating the data directly. Now, I'd like to talk about why a common vocabulary and structure is important. And to illustrate that, I was going to look at 4 examples that I've been involved in. I'm not the experimenter. These are data sets that I helped annotate. The first one is the Sternberg working memory from Scott's lab. And the events were coded with a single value column. There are a large number of codes, of which you see a few of them. And this study was chosen as part of the EEG Mini Labs replication study. Another study from his lab, the experimenter used 2 columns to encode events, and you notice that it's not very understandable what they are, but you needed both columns in order to determine what kind of event there were. Another example is an driving experiment with 4-digit event codes, and the position of the digit determines something about the meaning of the event. And these same experimenters did some additional experiments, and they needed 5 digits to actually encode all the cases that they wanted to document in their experiment. And none of these, if anyone looks at this data, would they know what these events mean? And so it's very important to have descriptions of what the codes mean and a common annotation framework so that tools can use the information downstream. So let's talk now about head integration in BIDS. So head is a standardized part of BIDS, and it's validated as part of BIDS validation. The head version is specified in the dataset description.json file of BIDS. And there are 2 use cases and approaches for annotating head in BIDS. One is to annotate each line of the event file individually, if you're annotating events. And this is useful for things like artifact identification. And the second is to annotate the unique codes that appear across the data set. And this is the sidecar approach. And you can use combinations of those approaches. And as I'll show you now, there are tools for supporting annotation using either approach. So let's start by looking at the structure of event files in BIDS. So event files are tab-separated values containing the markers for things that happened during the experiment. Each row represents an event marker. The columns have information of a particular type about the event. And BIDS requires onset and duration columns, and you can add other columns as desired. You don't have to use trial type and value columns. These are not required in BIDS, and there's a variety of columns that are used in event files. So it's important not only to identify the codes that are used in a given column, but to identify what kind of columns there are, because they're all different. So BIDS uses a JSON sidecar mechanism to provide metadata about what these columns and their values mean in BIDS. And a JSON sidecar, it's a dictionary. The keys of the dictionary are the names of the columns. And here's an example. The head annotations are in the 2nd level of the dictionary. And they're at the same level with the word levels, which are column descriptions of the individual unique values in the columns. And the head annotation workflow is you start with your event file and you create a JSON file with the unique event codes for a given column, and this might be done for multiple columns. And usually you just need one of these JSON files to describe all the kinds of events in your file. And these are the things you're going to annotate by providing head annotations for. And you fill in the head annotations for those unique codes. And you're done as far as annotation goes. At analysis time, the head tools will take the values in the event file, the head annotations that might be from the events file, which are used to annotate individual event markers, and you combine them with the annotations associated with the event code unique values that appear in each row of the event file. So it's assembled and provides a complete assembled event annotation for each marker, which is then used by downstream tools. And one of the important things to know is that BIDS allows head annotations not only in event files, but in many of the different kinds of BIDS tabular files using the same mechanism. And so this is a way of getting other kinds of metadata into your experimental timeline and your analysis tools. So one of the things that you needed to do there was generate a JSON sidecar. And we have head online tools to generate a template file from an event file. And here's an example on the right illustrating that. So you select the action that you want and the event file you're going to create a template from, and you pick the columns you want to annotate and which ones are going to be annotated individually rather than as a whole. You press process and you get a JSON sidecar out. And the JSON sidecar looks like this. And you see that then you can fill, this just has dummy head annotations. They validate, but they're not all that interesting or informative. And you fill in the descriptions of your events and your head tools. Usually you only need one of these JSON files for the whole data collection, data recording collection, and you're done. Now I'd like to turn to the head mechanisms for event context. So here's an example of an experimental timeline. And if you look at this event marker A, it has a head annotation, which is a duration 3 seconds, a label A. And that was constructed because there was a duration column there, and that had the 3 seconds. And what that tells head tools is that A is in effect not only at the starting point, which is at a point event, but it extends all the way through the end of this oval 3 seconds later. And the point is that at any intermediate point, event type A is in effect, and that means A is providing a context at the experimental timeline. So if this one black dot, in fact, A, B, and C are all ongoing. And also, you'll notice that head has a mechanism for mapping applicable metadata. And so if you've annotated using head some of the other metadata in your BIDS data set, that can be a virtual event that extends over the whole duration of the timeline and be incorporated into a single head annotation. Now, here's an example of some of the downstream tools that are used, and this is from the head remodeling tools, which are, here's an example on the left. So the idea is you use head tools without programming by specifying a list of commands in the JSON file. And so this JSON file has 2 commands. It has something that validates all the event files in the BIDS data set. And then the second command is to produce a summary of the tags. And this can be applied to the entire data set or to a single file. And you'll see the result of this. It goes through and figures out all of the annotations for the events markers in the entire data set and gives a summary of what things are present. Now, you'll notice that in this summarized event tag, a couple of things about it. The validate is fairly simple. You just have to give the command name, but the summarize tags allows you to specify a template where you give the titles that you want the event summary types to come out on. So the sensory event is going to organize all of the counts for tags that are listed here and their children, and it'll be presented under the sensory event title. Now, you also notice that there's an include context true. And what that means is that rather than treating the annotations as occurring at a single point at which they occur in the BIDS annotation, if they have temporal extent, that is, they have a duration or are given with an onset and an offset, then all of the event markers at the intermediate points will count that tag as being in effect there. And so this allows you to map context. It also has capabilities of extracting design matrices based on the condition variables. And it has a number of visualization tools, such as generating a word cloud based on these counts.
Hi everyone, my name is Young Truong and I'm a research engineer working at the Schwartz Center at UCSD with Scott Makeig, and I'm also collaborating with Kay on HEAD. And so I will be giving a tutorial on our annotation tools. There are two main tools that we developed to help people familiarize with HEAD, as well as help them with the annotation process: the HEAD schema browser and the WebC tagger. I will go over quickly the HEAD schema browser, but you also will see the schema browser in the WebC tagger as well when we get to that section. And throughout the tutorial, please feel free to open up any of these URL and follow with me throughout the tutorial if you want to do it more hands-on. So here is the HEAD schema browser. So this browser allows you to view any node in the schema in this hierarchical structure. And as you hover through any tags in the schema, you can see their information being displayed on the box on the right-hand side. So there'll be the description to the node and then also the properties attached to these nodes. You can collapse and expand any subtree of the schema to look for more detailed tags. As well as you can search for any specific terms in the schema using the search box above. So for example, if I look for participant and click on the participant response tag, it will scroll down to the tag in the tree, and then you can read more about it as well. Click the scroll to the top button to bring up to the top, and you can switch to other schema. With HEAD, we also have library schema to extend the HEAD vocabulary to more specialty subfield. So for example, the SCORE library schema contains terminology to describe clinical annotation as well as EEG artifacts. And this schema browser is very helpful as well. And we have integrated into the annotation tool ctagger, which I will show next. And again, you can open the URL ctagger.head.tools to follow the tutorials if you so choose. So here is the landing page of ctagger on the web. You can start by upload a BIS style event JSON or TSV file. For this tutorial, you can just load the example event.json file that we have already prepared for you for some simple annotation quick starts. And once you load a BIS events file, then you will see the drop-down menus. The top one is for you to select different columns in the events file. And then once you select a column, then the second drop-down menu will let you choose different unique values in that column. So here in our experiment, there are two types of events. There are the squares events, which display an image of a green square on the screen. And then there's our RT event that indicate subject button pressed whenever they think there's a target appear on the screen. As you select different column value, then CTAG will pull out the description of that column value, as well as any head annotation might have already been associated with those values. So if you scroll down to the area below, here you see the full view of the event.json file content in the bits format. So as you can see here, we already have annotated the square event types for you as an example. And we have prepared this annotation following the guide, the head annotation quick start guide, which is available on the web as well. So if you click on this link, it will bring up this page, a documentation that we have prepared. We have built a simple recipe for head annotation for you. One features that we are developing is the suggested tag features. So it's still under development. What we are using here are ChatGPT agents, as well as the head validator. Two agents, we use the multi-agent agentic AI approach to help suggest tags from the schema correspond to the description of the event type. So for example, here, I'm going to try to suggest tags for this subject button press events. And so here is our final tag suggestions. And you can see below as well, like the conversation with the ChatGPT agent that has been occurred to generate this tag. So it started with some suggestions. It passed the annotation suggestion to the head validator. And now we found some invalid tags. And now the GPT agent correct itself to produce a valid tag. And then we convert all the final annotation into the short form for easy reading. Now, our final tag suggestion, you can copy this, and then you can add it to your annotation as well. So this is a helpful feature, and we are continuously developing it and modifying it so that it will provide tags that are relevant as well as detailed for the description. Once you finish your annotation, you can save this event.json file and place it into your bits directory. And as Kate mentioned earlier, the way this works is because of the inheritance principle. If all your events file share the same structure, you can prepare a single event.json file and place it at the top level of your bits directory. And that's the single file that you need to prepare. And the annotation and the description will be applied to all the downstream events TSV file in your bits dataset. A quick example of what it will look like in analysis time if you have a head annotation would be, so if you now upload a example events.tsv file that corresponds to the same task, in analysis time, the head tools will assemble the annotation for each row of the events file, giving your annotation in the event.json file. And so now for each row, each event in the events file, you will have the full annotation of that event row. And then you can do ad hoc extraction based on looking for specific tags. So for example, if this data set you want to extract out visual presentation event, you can search for a visual presentation tag, and it will extract out all the square events for you. And that's mainly it for the annotation tools that we have developed and make available on the web. Please feel free to check them out and start annotating your data set. So besides the schema browser and the web tagger, we have also developed other tools to help with other aspects of working with head, including the Python tools available as head tools on PyPy, and this is the main functionality in our code base. The online tools are also available on headtools.org. It is a series of functionality that you can use for processing your events file, event JSON sidecar, or individual single head string. It can also help with the schema development process, and all the code can also be available on the head web repo on GitHub. We have covered the C tagger. It also is integrated into EEG Lab head tools plugin, so you can call the C tagger via EEG Lab interface. And then we have the JavaScript validator that helps with the validation of head tags in bits dataset on OpenNeuro or other bit supporting platform. And finally, we have the head tools in MATLAB, either as an EEG Lab plugin or as a service, as a MATLAB library that any other analysis tool that want to support head can use our features and functionalities in their tools. all our tools are open source, and you can see our code base on GitHub, and feel free to make suggestions, post issues on GitHub, and make use of them. So now I want to invite Scott back to have some closing thoughts.
So I hope that you've learned in this 45-minute presentation the importance of HED going forward for open science, informatics, for time series neuroimaging data, including EEG, MEG, fMRI, intracranial EEG, or other modalities that record time series information. On this screen, you see the links to sources of information about using HEAD, about all its documentation, all about its tools, including its new C-Tagger tool that will continue to be improved, that will help you, first of all, play with it to learn how head annotation can fit into your workflow, and then use it to help you build analysis-ready data sets that you can share openly and thereby contribute not only to the future of neuroscience, but I argue to your own career in neuroscience by creating interactions, generating citations, and contributing to the forward progress in the whole field. If you want to know more, or if you want to contribute in any way, please write to the head maintainers at gmail.com. Thank you.